Oh, I have to shut up. Hi, my name is Laura Nickerson.
And I'm Mr. Radford.
And we are Ms. Nixon, and we are here to tell you guys about the scientific method. We're going to go through the steps of it and give you some examples, and you should be able to reproduce the scientific method and understand the different steps. So the first thing you have to do is ask a question, and with that comes a lot of planning.
Okay, and so the very first part of that is making observations, and this is something that we don't just do at the beginning.
No.
We make observations throughout the entire scientific method process, and so we want to be as accurate as possible, as detailed as possible.
Yeah. Don't want to make up stuff. Just write down what's happened, and that's a good science. You're going to use all your different senses. So a lot of people think, well, it's just my sight that I'm going to use in the lab, but you can learn a lot of things by listening and sometimes tasting, not always. But you want to use all your senses to gather those observations so that you can have a good experiment. There's two different types of observations. The first is quantitative, and that means these are exact things that no matter who observed it, they would all get the same observation. So, for example, how tall is a plant? That's not going to change whether I look at it or Mr. Radford looks at it.
The next are qualitative, and these are more descriptive. These can be somewhat subjective at times. If a person has maybe a better view, the lighting changes, whatever.
Also, opinion kind of comes into this.
It can.
If I might say the plant is like moss-colored, and you might say it's yellowish colored. So these aren't as exact, but they're still important observations. This leads us to a research question. So, for example, we may be doing an experiment with plants. How does the amount of light a plant gets affect how a plant grows? That's what we're going to try to figure out, something we don't know the answer to.
Okay, and so this leads us to kind of we want to make sure that we narrow our focus. We're not testing other things that we don't want to find in our experiment. So that we have these are called variables. And so we want to limit the number of variables, and there are three main types.
The first type is the independent variable. This is, what do we want to change? This is like what we're actually doing the experiment on. So in this case, we're going to probably change the amount of light, because that's what we want to see. How does light affect a plant's growth? An independent variable. Okay.
Then the next type is a dependent variable. And this is the variable that we're basically kind of testing. So what our result is going to be.
How tall does that plant actually get?
Yeah.
And then finally, we've got the constant variables. These are the things that we don't want to change no matter how done the experiment. So that later on, we know that our results are based upon how much light the plant got, not on how much water it got or how much fertilizer it got. These things all have to be the same no matter what plant we're testing throughout the whole experiment. They're constant.
Okay, and we've given these examples. Independent variable is the amount of time the plant receives the light. Dependent variable, how tall the plant grows. And the constant is the amount of water, temperature, type of soil, species, everything that we want to keep the same, except for what we're testing.
We expect you guys to probably list out these three variables before you start your experiment, not afterwards, because it really helps you plan out what you're going to do. That leads us to our next step. We've asked a question, we know what we're doing, done a little background, and now it's time for us to construct a hypothesis.
Hypothesis is basically an educated guess. I mean, it's an answer to our question. And we always want to form our hypothesis in an if, then, because format. That way, when we're done, we can go back and say, yes, I was right, or, man, I was off.
So in our plant example,
Tracy Reed, you could call or stop by the front office, Tracy Reed.
In our plant example, it would be, if I give my plant 1 hour of light a day, it won't grow as tall because plants need light for photosynthesis. I have if, then, because, all included in my hypothesis. So what's next?
Well, hey, let's start testing.
Let's test it. The favorite part of science. Let's do the experiment. And with that comes collecting data. I don't know how many times I've had kids finish an experiment and I'll ask, what happened? And they're like, oh, I don't know. I didn't write anything down. I'm like, you're going to have to do it again. Yeah. So we really have to collect the data because you can't remember everything that happens in an experiment from one day to the next.
Okay, so generally in an experiment, we have our control group that we collect our data from, and we use that to compare it to our experimental group.
That way we have something as a baseline that we can say, well, our experiment really did have an effect, because otherwise, if we don't have something to compare it to, we can't prove it's what we adjusted that had any effect. So, for example, if we were doing this plant experiment, we want to have a group of plants that didn't get any sunlight at all. So then we could show that the amount of light did have an effect. It wasn't something else and not light. So now that we got everything, we're running the experiment. How should we write this information down? Should I just try to remember it? How am I going to make sure that I have all the data for the next day? I probably want to take some collection methods for recording my results.
And good scientists are meticulous in recording their information.
Again, I don't know how many times I've looked at students' results, and I'm like, what is this number? And they go, I don't remember which number that was. Was that this? Was that that? I didn't label it. So you probably want to design some type of table or graph before you start. So then it's just a matter of filling in your data, and it's all organized from the beginning.
If you notice on the chart too, it has the units. So we know that average growth in 1 week is measured in centimeters. If you don't put that, we don't know what those numbers mean.
And you may not remember, so it's important to write it down. We expect your data to be neat because it's an important part of the experiment. So when you make your tables or your graphs, use a ruler. Make sure you have it scaled correctly. Give it a title so that we're not looking at. And like Mr. Radford said, we need lots of labels of measuring, labeling your graph, and then we just put our data in there, and we don't fake data. Whatever happens is what happens. It's an experiment. You're never going to get marked down in class because your data didn't turn out right, as long as you were recording what actually happened.
Exactly. And that's one great thing about science. If we mess up, that's okay.
Maybe that leads us to an even bigger discovery than what we originally started that way. All right, so there's lots of different ways you can present your data. We love graphs. I just love math too. So a line graph is appropriate for some things, but not everything. Sometimes you might need a pie graph that's going to better represent your data, or sometimes it might be a bar graph, or even maybe just a table that's not graph. And you can also include your data in paragraph form, because some of your quantitative and qualitative data isn't always going to graph out.
It's true. So graphing, you want to make sure you have the X and the Y axis labeled. Usually the X is labeled as the independent variable, the Y is the dependent. Make sure it's to scale. It's always frustrating when students make their graphs really small. And use the whole page, make it look nice. Show off what you measured and what you learned. Make sure you have the units labeled, plot your points, and give a description or to your title.
All right. So the last part, I think, is my favorite part to read when I read through different experiments, and that's the conclusion. What's it called?
This is the meat and the potatoes.
This is
where all the main substance comes from.
This is where we as teachers can read and see if you really understood the experiment and what kind of results you got from this experiment. So it has to be based on your results and what you think those results mean. Results and reasoning is how that's called. And there's a few steps you need to take.
So you make sure you restate your purpose of kind of what you were doing, summarize your procedures, you look at your results and you restate the numbers, any trends that you see throughout that. Hey, and got to show off your scientific vocabulary.
That thing grew this much, this way. What is that? You got to use the words from the lesson that we're talking about so that we know that you understand them. Make sure if you made mistakes or you think they were mistakes, you talk about them. This is why I think my data is not as I expected or is higher than everyone else's in class. And then I always like to see improvement. If I was to do this experiment again, here's what I would do differently to make my data better.
Make suggestions to future researchers, and then also, hey, what new things did I learn from this? What new questions do I have? Because science is an ongoing process. One scientist comes up with an experiment, leads to new questions. Someone else takes that and retests and does new things with it.
And speaking of other scientists, another scientist anywhere in the world should be able to read just your conclusion and understand your whole experiment, how to do it again, how to replicate your results, and the things you did right and wrong in it. So your conclusion is definitely the meat and potatoes of your lab. Well, we hope you guys enjoyed our notes section today. We'll be doing an experiment in class to practice these steps with scientific investigation. Make sure you fill out your note sheet and have it ready to turn in to class. And thank you for joining us on the Viking Science Academy.
We'll see ya.
10 minutes. That's better than.
Okay, yeah, that video I did take. Looks like it's still going.
Yeah.